So our immune system has an enormous task. It has to recognize and respond to the myriad pathogens that we encounter throughout life, whether they're viruses, bacteria, fungi, parasites, worms. The immune system has to respond and do so with exquisite specificity and generate memory responses so that if we re-encounter the same pathogen, which is likely to be in our environment locally, we're not going to get sick again, but we're going to respond very quickly and fight off the infection. So that's a big task, right? But there's an arms race. So our pathogens are constantly evolving to evade immune recognition. And this means that throughout our lives, we have to continue to generate new immune responses. And a major component of our adaptive immune system, the part of immunity that responds very specifically to pathogens, to a particular pathogen, like this year's SARS-CoV-2 strain or seasonal influenza, and remembers it so that we don't get sick from it again, is our T cell. And T cells are amazing. They can do a whole variety of different things. They can directly kill infected cells. They can stimulate other cells in your immune system to secrete inflammatory molecules or antibodies that provide protection. They can even kill cancer cells. But that's a lot to ask of one cell type, right? We're asking it to do all of these functions and very specifically recognize all of these different pathogens. So how is it possible that T cells can do this? So the key to the specificity of T cell recognition is that they all express a T cell receptor, a protein schematized here with our purple receptor sticking off of the T cell. And the idea is that if you have an infected cell, like the virally infected cell depicted here, T cells can come along, and if they have a T cell receptor with a lock and key fit for that pathogen, so the infected cell is showing a little piece of the pathogen to the T cell, if there's a lock and key fit, that T cell will become activated, and it has the potential to kill off the infected cell. So that means that for each virus that we encounter, that we become infected with, there's a T cell with a different specific T cell receptor for this year's flu strain versus next year's flu strain, or any bacterial infection. That raises a new question. Is it really possible that we can generate enough T cells with enough different T cell receptors to recognize any pathogen out there? And so this raises an interesting question. And I'm going to ask you guys, how many genes do you think are encoded in our DNA? How many protein coding genes do you think are encoded in our DNA? Any guess is good. 100? That's one guess. Anybody else? This is the blueprint of life. It's going to encode all of the genes that make up all of the specific cell types in our bodies, our neurons, right? The genes are encoding proteins, the building blocks, the machinery of our cells, our neurons, our retinal cells, our endothelial cells that make up our vasculature. How many genes do you think it would take in all of our DNA to carry out all of these distinct functions? A lot. I got a lot. So I think, when I was thinking about this as a student, I would have thought, well, probably this is going to take millions of genes, right? It turns out that we have about 23,000 protein coding genes in our genome. That's not really a very big number, right? It's kind of small because not all of these 23,000 genes are encoding distinct T cell receptors that can fight off pathogens, right? Some of them are encoding retinal proteins in our eyes. Some of them are encoding insulin that is made by our pancreas. Okay, so is that really enough to make all the different T cell receptors out there? No. In fact, the number of different T cell receptors that can be generated by one individual is on the order of 10 to the 18th different T cell receptors. So that's a really big number, right? And this raises a conundrum. If we only have 23,000 genes, how on earth do we encode in our DNA 10 to the 18th different T cell receptors? So when you take biology, you learn that, your DNA encodes a gene. That gene gets transcribed into a message that's made of RNA, and that RNA gets translated into protein, the machinery, the building blocks of our cells. But that's not the way it works for T cells and the T cell receptor. Instead, we have many diverse gene segments that encode parts of a T cell receptor. And as T cells develop, they literally chop up, cleave the DNA that's in their nucleus, and they bring together these gene segments of the T cell receptor in this combinatorial and very imprecise fashion, randomly adding pieces of DNA in between the segments. And that's how we're able to get to 10 to the 18th different T cell receptors, which is awesome for pathogen recognition, but it raises a real problem. These receptors are generated randomly, and that means that by chance, some of them are going to recognize our own proteins. And we don't want T cells fighting off our own healthy cells, right? Because that's autoimmunity, that causes autoimmunity. We want them to fight off pathogens that we encounter. So for a long time, I've really been fascinated with how do T cells develop and become educated to fight all the pathogens we encounter throughout life, including new emerging pathogens like SARS-CoV-2, the causative agent of COVID-19, while simultaneously maintaining tolerance to our own bodies. So T cells develop in a specialized organ called the thymus, depicted here. There are two major regions of the thymus. The thymus's sole job is to make T cells, and it's doing this throughout our lives. So the thymus actually sits in our chest cavity just over our heart, and again, its function is to make T cells and to educate them to be self-tolerant. So the outer region, the red part of the thymus there, is where the early cells come in that haven't yet decided if they're going to become a T cell. If they get cues from that environment that say, okay, you're in the right place, you're going to become a T cell, they start chopping up their DNA and rearranging it to make a T cell receptor. Now that's a pretty kind of scary and dangerous process. It doesn't work in a good number of cells, and many of them die. But for the few that actually make it and do express a functional T cell receptor, they're now going to move into the central region, the medulla, the central region in green of the thymus, and this is where they're going to learn to be self-tolerant. So let's look at that process, which really fascinates me a little more. So the idea, again, is that you have a T cell with its T cell receptor here, and if it's tested it and it's functional, it's going to move into the medulla. And these cells are going to start differentiating into the different types of T cells that can kill virally infected cells or help produce antibodies. And in the medulla of the thymus, they're going to encounter two other key cell types, thymic epithelial cells in blue and dendritic cells in green. And what those cells are doing is they're presenting pieces of our own proteins, like a little display library, to the developing T cells, so that if that new T cell receptor has a lock and key fit for our own proteins, it'll get a signal that it's autoreactive, and it will undergo cell death. It'll die and be eliminated before maturing as a T cell. So this is a key process called central tolerance that helps to ensure that our T cells won't attack us. But this raises another interesting question, right? So, the proteins that a thymic epithelial cell expresses should be, proteins that are important for being a thymic epithelial cell. So great, it's going to show pieces of thymic epithelial cell proteins to developing T cells, and we'll be tolerant to those. Same thing for dendritic cells. They'll show dendritic cell proteins to T cells. But once T cells get out into your circulation, they're going all over the body, right? Into your CNS, into your big toe, into your pancreas, into your heart, scanning around to see whether or not there are pathogens present. So you don't want your cells to be tolerant only to what happens to be the proteins expressed by an epithelial cell or what happen to be the proteins expressed by a dendritic cell. You want your T cells to be broadly tolerant to your own body, right? So how do we accomplish that? So there's a couple of cool mechanisms in the thymus that are specialized for generating broad self-tolerance. First, these thymic epithelial cells actually can express about 90% of all of our proteins. So they have a specialized mechanism for doing that, and we know that that's very important for tolerance because people who have problems with that mechanism have multi-organ autoimmunity. So it's very key that those thymic epithelial cells express all of those proteins. Well, what about your dendritic cells? They can acquire proteins from your blood, from circulation, and present it to T cells to get rid of autoreactive cells. They can actually start to other tissues and traffic proteins from those tissues into the thymus to present And this means that there's this very broad display of self-proteins in the thymus that are kind of sparsely distributed around that central region. And the thymocytes, which are shown here in purple, have to really rapidly run around and find all of these, test their T cell receptors against all of these dendritic cells that are shown in yellow here, and thymic epithelial cells to see if their TCR has a lock and key fit for self-proteins, and if so, then they undergo tolerance. And so we're really excited about understanding the mechanisms that govern entry of thymocytes into the thymus and rapid scanning of all the self-antigens to ensure self-tolerance. But there are some other things that we learn from studying autoimmune disorders that have been really intriguing and more recently recognized. And I want to kind of point you guys to some of those. So you would think that if someone, so first I should say this process isn't perfect, right? The thymocytes have a limited period of, the developing T cells have a limited period of time in the thymus to test their T cell receptor for self-recognition. And we know that there are about 50 million Americans who have an autoimmune disorder. So this process doesn't always go right. It's about this is the third most common disease category behind cancer and heart disease. So there are a lot of people who have issues with this process and generate autoimmune responses against their own cells. And in looking at that, there were some things that were interesting. So you would think that people who have autoimmunity, where their immune system is sort of ramped up, right, and it's really going, there's lots of inflammation, that those individuals would not be terribly susceptible to infectious disease because they have a sort of hyperactivated immune system. But instead, we've learned that autoimmunity and immunodeficiencies, susceptibility to pathogens, often go hand in hand. And that was really confusing for a long time. And recently, during the COVID-19 pandemic, when we had the ability to use all of our modern tools to really study what immune responses looked like in individuals who had asymptomatic infections versus those who went on and ended up being hospitalized from COVID-19, some of whom ended up in the ICU or succumbed to infection. We were able to look at what was different about their immune response. And one really interesting thing that came out was that people who ended up in the ICU with COVID-19 tended to have a high frequency of autoantibodies, so an autoimmune response, against the very proteins that our immune system makes to help fight a viral infection. So we were mounting these individuals were mounting autoimmune responses against their immune system, which prevented it from fighting off the virus well. So this is why you had immunodeficiency and autoimmunity in a lot of cases at the same time. And this brings up another interesting point. When we have an infection, our body exists in different states. When we're kind of at rest, at homeostasis, when we're healthy, versus when we're infected, and we all kind of empirically know that, right? You feel bad, you have fever, things have changed. You're expressing different genes and therefore different proteins when you're sick. So how do we make sure that our immune system is tolerant to the proteins that are expressed during infection? And this is cool. So one of the things that we and others have found is that within your thymus, some subset of the dendritic cells actually look like they're in a state of infection. They're like this at steady state, even when there's no infection or pathogen present. And that's really important for presenting to the developing T cells these self-antigens that they're going to encounter when your body is in a state of infection. Okay, so that's just giving you a little bit of a taste of some of the things I think are really intriguing about T cell development and selection. And I want to take this back to your theme of forward, right? So as we're learning all of these things about the immune system and about central tolerance, what can we do with this new knowledge to help prevent and treat autoimmunity and to understand better the causes of these autoimmune disorders? And for some of you who are out there, this will be really relevant to you as budding scientists. And for some of you, you can think of this more as a rubric of how to think about any problem that you encounter in life that you think is important and something you want to tackle. First, use your curiosity. Ask questions. When you see things that don't make sense in your textbook, in your class, in life, right? Patients who have both autoimmunity and immunodeficiency, why would those go hand in hand? Start thinking about that and realize that if you can't get a satisfactory answer, find some good information about it, it's a really good question, right? So that's a good jumping off place to do some research. Then be creative in thinking about how you could address, creative in generating models of what might be going on to address your question of interest, and creative in generating, in my case, experiments, right? And coming up with experiments to test those new models. And then this is a really key point, be flexible in your thinking. So when you start to get data back, right, answers back to your questions, and you look at what's going on, sometimes the data, you've got a great model. You had this wonderful idea. And sometimes the data don't fit your great idea. This happens a lot, right? So instead of just sticking to it and being stubborn, learn from the data, learn what it's telling you, and revise your model to fit what the information that's out there has actually showed you. And that's really important. A lot of times I think people think scientists are wishy-washy, they change their advice. You saw this a lot during the pandemic, they change their opinion. But in reality, what they're doing is they're learning from new information, and they're adjusting their models and their hypotheses to fit the data, and then generating new experiments to test the new models. And that's a key part of science. And finally, be inventive, right? Once you really have a handle on some process, in my case, disease processes, think about how you could use that new knowledge to invent interventions that will help patients going forward. Okay, thank you.